screen?
Yes.
All right. So, good morning, everyone. I'm Rebecca Wadham, and this is the last, I believe, in the Cross BRC Tick webinar series. Today, we're going to discuss SNP and RNA-Seq analysis that you could do in the Bacterial and Viral Bioinformatics Resource Center, which we call BV-BRC. BV-BRC is a recent combination of the bacterial BRC, known as Patrick, and the two viral BRCs, which are IRD and Viper. As I said, this is the last in the series, and this has been ongoing since March. If you missed anything, all the videos, all of the webinars are available at our BRC or at our sister BRC, ViewPath DB, where you registered for the series. ViewPath hosts the tick and the eukaryotic pathogen data. And I'll make all the slides available and stuff if you want to see those or not. If you've watched any of my webinars in this series, you know, let me close this up because it's distracting. You know that I take a paper and then we walk through the analysis of it, and then I say, what can we do? Could we do the same thing in BV-BRC? So this is today's example, and for those of you who are tick researchers, you're probably looking at this and saying, Borrelia recurrensis? Borrelia burgdorferi is transmitted by ticks, but recurrensis, I'm not so sure. Well, you're right. For the SNP analysis that I'm doing today, there are two requirements. Genome, which is easy to get, but also reads. This particular pipeline that we have in BV-BRC uses reads against a reference genome. And where I could find many papers, there was an excellent one on Borrelia burgdorferi that I really wanted to use, doing a SNP analysis, but they were only in contigs. And to find something that had freely available data that were in reads, I had to step up a bit. And still, this is an interesting paper, and I think you'll like it. And then you can use it as a model for your own analysis using tick pathogen. Come on. So, there we go. Borrelia recurrensis is the causative agent of louse-borne relapsing fever. So while it's not transmitted by ticks, it is transmitted by lice, specifically the human body louse, Pediculus humanus. This particular study looked at 6 isolates that were cultured in different years in Africa. And this was the first study studying the genomic variability of this particular pathogen, of which still there's only one genome in the public resources. So they provided us with the sample metadata of their different samples and the years that they were isolated, and you can see they were in Ethiopia and Somalia. They mapped the reads. Because these were taken from swabs from human samples, when they sequenced the DNA, they were able to map the genomes, map the reads to Homo sapiens, and they also mapped them to Borrelia recurrensis. And they used the variant calling pipeline that they have in the CLC Genomics workbench called basic variant detection. They also assembled the genomes, and they did those using the CLC Genomics workbench and SPADE. Strangely enough, even though I know they assembled them, they don't seem to have put them in GenBank. They have the reads in SRA, but the genomes themselves are not in GenBank. And they generated phylogenetic trees using SNP and maximum likelihood trees. And I liked this a lot. They showed where the SNPs occurred within the Borrelia recurrensis A1 genome by mapping the reads to that and then showing, and you can see in this picture which of those shared which particular SNPs. So And this is just to the first chromosome, actually, or the chromosome, let's put it this way. This particular genome has 1 chromosome and 6 plasmids, but they just were looking at that. So how much of this analysis can be done in the BV-BRC? So I'm going to leave the slides for a minute, and I don't want to leave you in the dark, but let's go over to BV-BRC. Now, we have 2 intense examples to do today, SNP analysis and RNA-Seq. If you've watched the webinars before, you know that we can do assembly in BV-BRC. We can build trees. I've stepped you through it. You can see in the other webinars, or you can see that in the tutorial. We can also, and we will show this in the second part, of looking at trimming and read quality and aligning to a particular genome. We're going to be doing that in the RNA-Seq part of this. But because we're in kind of a tight time frame here, we're going to go straight to the variation service. So you'd click on Tools and Services, and then you would click on the variation analysis. And we're going to go right here to do this analysis of these reads. Now in BV-BRC, you could upload your reads to compare them. So if you had them on your computer, you could click on that file and the file folder, and then you could upload reads here. Or you could do the same thing with single reads. Or if you have reads in your workspace, you could search for them here with an identifier that you might have, or you could see which ones you uploaded most recently by clicking on that. But for this example, we're going to go with these SRR numbers that this study originally used. And so I'm just going to cut and paste this so you don't have to watch me misstep things. So, and you can see that I've got a Word document over there because it shows that. So this is one of the SRR numbers. And I move that over. And once I move it over, I had to click on this arrow to move it into the selected libraries. And when I click on the information icon, you can see that it's already gone to SRA, and it has some information about this particular read file. And so I'm just going to go in and add them all over because there were 6 of them. And yeah, oops. I got the wrong one there. I don't want that one. This guy. See, it's a good, yes. You see this? I was going in the wrong direction, and this is a occlutus thing. I don't want that, so I just click on this. So I've taught myself an important lesson, and you're seeing it too. I knew that this went up, so it's always good to check. So we're still at Borrelia recurrensis. 8, 9, and, well, actually that's not 8, 9, this is 6. So yeah, so we've got all the strains here, all 6 strains. So we're ready to go, but we still can't submit yet because we have to figure out, we have to assign an aligner and a SNP caller. So to see the aligner, you can click down, and we have 3 different flavors: BWA Mem, Mem Strict, Bowtie 2, and Last. Last is for long reads. The others are for short reads, but I'm just going to stick with the default right now, although I run all of them so I can make comparisons. Of course, not Last if these are short reads. And then for the SNP callers, we have Freebase and SAM tools, so we'll stick with that. And let me point out that if you want more information about the service, you could click up here for the tutorial. And that'll open another window that tells you about these different services, and it gives you details about the different aligners. and about the different SNP colors and when you would use them. And it also, at the bottom, gives you the references, because, you know, I'm a research biologist too. I know that I do these things, and then like 6 months later when I'm writing the paper, oh, what did I do? I can come back here and reference these things here, so it's all there for you. Okay, so we've got the aligner and the collar. Now we need the reference genome, and this was Borrelia recur, and I'm gonna pause here hoping, yes, because I typed terribly. So you can see that if it's got this little lock in front of it, that means it's my private genome. These are all the instances in the database of this genome, and you notice they're all A1. We have the entire genome here, which is the representative, that's the chromosome and all its plasmids, but they also separated it out and have each of the plasmids here so you can compare to each of them too. So I'm going to click that one, and now we have to, so it's going to align the reads to this genome, and now we have to create a folder to store the data in. If you had one created previously, you could just type on it or click on the drop down arrow, not error, but let's create a new folder. So I'll click on that, and up here I can click on a folder and I'll call it Tick Webinar 14 June. I have lots of folders. And then if I want to see the one I most recently created, because like I said, I have lots of folders, I just click on create it. It'll take me to the earliest one, so you can see I've been using this since 2015. But here's, and then click on it again, it shows me my most recent, and that's the one I want. And that's where the job will be stored, Borrelia A1. Whoops, A1 analysis, 14 June. And I always put a lot of information in, because as you can see here under jobs, I've run a lot of jobs, and I come back sometimes months later to look at these things, and I want to be sure that I can find it quickly. So when everything's filled in, like the submit button turns blue, I can click on submit. It tells me it's submitting my job, and then it says it's been successfully submitted. So I can monitor the progress by clicking on the jobs icon here, which will tell me how many jobs I have queued, running, completed, or I can go up into Workspace and click on my jobs, and it shows me that this has been submitted. Although it does say running, I never consider it to be running until I can see that the start has been filled in. Right now it's probably in the queue. Now these variation jobs run very quickly, but we're not going to sit around and wait for it to run. So what I want...
Rebecca, can I ask you a question before you go on? So
yeah.
With the read files that you got from SRA, you did not have to indicate whether they're paired-ended or single-ended. So
That's
the
right.
workflow determines that on its own. And
yes, we have an well, it's also was it this example or another example? I'm trying to remember, because I have two examples today. And this is always an important thing to take into account. When a researcher is getting ready to publish, absolutely the very last thing they do is they submit their data to GenBank or SRA, because they have to, and because we know I'm that way too, terrified that I'm going to get scooped, so I wait till the last minute. And because it's the last minute, sometimes there are mistakes made, and you don't care as much because you're trying to get your paper out, and doesn't matter, and sometimes the metadata is wrong. And it might have been this example, or it might have been the other one, I can't remember which one. They entered it as paired read. In fact, we could see, because let's see, it's hard for me with that thing up there. Here it is. So they entered it, so I could go here. Come here, you. Yeah. Come on. Don't be shy. Little SRA. Yeah, I think that this was entered. The metadata indicated that it was pair read, but it was single read. So
Okay.
there's some. So once we figured that out, we downloaded the reads. It might have been the other one though. I can't remember which of the two today. We downloaded the reads, and then it was fine once we stepped away from their metadata. So
So there, it's basically going to use the metadata, and obviously the assumption is that the metadata is correct. But if it's
yeah,
incorrect, then you might get a failure in the
I
run,
have
right?
been
Okay.
up to my eyeballs in SRA data lately, just for this and for other examples,
Right.
and
Right.
I'm often astounded by the lack of care when people are submitting their data. And
So
I've
that's an important point to everybody. If you submit data to SRA, make sure you're careful with labeling the metadata
yeah.
appropriately. Anyway, sorry to interrupt, Rebecca.
That's
You
okay.
can
I've
carry on.
had
I
to
was just
go
curious.
to, but in pursuing these examples, I've had to go to authors and say, you said, it says in your paper that the data was submitted under this, but that's, when I go to that number, it's something different. And they
Right.
always come back with, oh, I had a graduate student do it. Nobody says, oh, I did it
They always
myself.
blame the graduate student, right?
That's right.
So
Okay.
for everybody else, if you have additional questions, obviously feel free to either type in the chat window or just unmute yourself and ask a question. You're welcome to do that. All
Yeah.
right. So
So let's see what this job looks like, and I've put it here for you. So let's go into public workspaces. Or, come on, BV-BRC. And then we migrate down to BV-BRC Workshop. I've put it in TIC webinars. And for this, we have the Snip analysis with Borrelia. So let's click on that. And I put the paper in here. I put different files in here. Originally, I was looking at FastQ alignments and the trees, but there isn't time to talk about those now today. Let's look at this job. So let's click on this, and this is the variation. And when it has this checkered flag, it means that that job successfully completed. So we click on that. And it comes back with a ton of different files. And we'll briefly walk through the main ones of those. But note how fast this ran once it ran. And you notice I put 6 read files in there. You can put as many as you want in to compare it. But when you do that, remember that you're going to have to analyze it. So if you have 100 things in there, Godspeed and good luck. So the first thing I want to show you is up here is the genome browser. So let's click on that. And that's taking me to the reference genome, which is this Borrelia recurrensis. Let's wait a moment for it to load. Now this is one of the few places in the resource where you could actually compare the annotation by BV-BRC. So if I mouse over something, it'll tell me what it's called there. The annotation in RefSeq. And I can, there are these many contigs, a chromosome and 6 plasmids that I could look at. And this is just showing me that I'm looking at this part of the genome. Now I can certainly drill in, but what I want to show you is these particular files are showing you where the SNPs are in the various read pairs, or the reads. So there's one, that's the second sample, that's the third sample, that's the fourth sample, that's the fifth sample, and that's the sixth sample. And let me stretch this out. I'm going to get as much real estate as possible so that you can see pretty easily where these reads are and how broadly they're shared at this high level. And I could zoom in to try to see where these genes are, and it's going to take several clicks to get there. You know what, I'm going to remove the RefSeq annotation at this point so we have a lot more real estate. And as I zoom in, I want to keep this in the center, and I'm going to include the reference sequence too. So as I keep zooming in and keep zooming in, don't be shy, little guy, you can see exactly where the SNP is occurring and in which frame that it's happening in. I'm going to click a few of these so you can see that we also allow you to bring in the BAM file so you can see how deep the reads are for that particular call. And when we talk about the different files that come with this job in a few minutes, the scores are based on the number of reads that map to this. And so, I mean, if you're interested in playing with these figures, you can, you know, say I want to hide the reads aligned to the forward strand, things like that, all the different kinds of things that you can do with that. You can mouse over things to see different values of different things here. So, but you know, this isn't, although this is an interesting thing to see, and I can see which gene it's in by looking at that, if I had to do the entire analysis based on the genome browser, I think I'd be very unhappy. So let's go back, and I want to point out scrolling down, that we have this particular file, which is the all variant HTML, which you can also download the the TSV file for that. And I should also point out the library's text file, which I can highlight and view. It tells me which of these samples are which. So I want to go back to that all variant HTML file and click on that and view it. Now you remember you can download it, but I love this service. And the main reason I love this service is this particular file here. It summarizes all the data, and it says that this particular sample had a SNP. And when you, in the reference genome, this is the contig it's on, this is the position it's in. In the reference genome, and let me move this over so we have all the real estate we need. In the reference genome, C is called there. In the variant, it's a T. All of these scores are related to those read files that I showed you when I put the BAM files in. And for depending upon the, you know, if you run BWA MEM or Bowtie 2, they'll each have slightly different scoring. That it's a non-synonymous change. The gene nucleotide goes from ATG to ATA, and this actually changes within the gene. It's showing you the change in position is at place 846 is where the change occurs, and this actually results in a change in the amino acids from a MET to that. And so it also tells you if it was a frame shift, it would tell you that. It gives you the gene ID. It gives you the RefSeq locus tag. This is the BV-BRC gene ID. It's our unique identifier. This is the RefSeq GenBank gene ID. It's their unique identifier. They are both pointing to the same gene. This is the gene name, its function, upstream, downstream genes, and it rates the SNP, tells you what it is, and tells you how bad it is or not. And if you were interested in one, like this is at position 389. If I wanted to see this gene, this is a hyperlink. I could click on that, and that would enter, that would go to the feature landing page within the Borrelia recurrensis A1 genome. And here is the particular gene. And this is all a bunch of information about this gene. You can do things like look at the neighborhood and stuff. But when I'm doing a SNP analysis, I always go to CDD because I want to see the domains there. And actually, you can now do that at BV-BRC. That's a new functionality. But remember, it was at 389 that we were getting this change in sequence. And that is right in the middle of this particular domain. So that's how you can drill down and do deeper analysis on that. So I want to go back to this and show you some other things that you can do with this. For example, I can click on for each of the individual read files. I can click on that particular one and click on view, and it's showing me the same information just for this particular sample. And I'm going to show you something. Remember how the authors, how they showed you, put the SNPs on the genome. So I'm going to show you how you can do that in BV-BRC. So I would click on this, and well, I can download it here. Also, I should point out that you can filter on the data here at this point. But let's download this thing. And then I always show in Finder. Oh, Finder's over here. Sorry, it's on my other screen. And I'll open that with Excel. Hopefully it'll open. Oh, this is the way I did it. This is the previous one that I was playing with before. So this is the information. Let me move this away. For this particular sample, which was the SE1 sample. And I'm going to increase the view here so you can see what I'm about to do. And I'm just going to show it to you so you guys can watch this later. But in order to view this, these SNPs, and upload them into a viewer that we can see in BV-BRC, there's a specific format that's needed for the viewer to read that. So I'm going to show you how you can do that. And for that, I don't need this particular column. So I'm going to delete this column, or I thought I was. Delete column. Oops, actually I needed that one. I deleted too many. Undo delete. That, I need that one. Okay, I don't need any of these others. So I need the contigs and I need the positions. So I delete these columns. All right, and then I'm going to temporarily create two columns, because what our viewer needs is the contig, and they need a start and a stop. And you may say, why can't I just use this 233? You can't use that because it'll be too small when you're looking at an entire genome. So you just need, we're just providing a pinpoint as to where this particular SNP is. So we need to make it big enough so that the viewer can resolve it and you can see it. And I actually like 1,000 base pairs or 2,000. Let's do 2,000 here. So I'll say equal to 233 minus 1,000. And yes, that's a negative number. We'll fix that. Equals 233 plus 1,000. Click on that. And then I'll go down and shift click and paste. I know it's probably, you're thinking, gosh, how boring is this? And yes, a little bit boring, but when we get to the end part, you're really going to be glad. I hope that you saw it. Now, when you see these negative values, I'm just going to highlight that so that I can easily see them with my eye while I'm trying to do, because I have to resolve those. Because if I give those negative values to the viewer will say, wait a minute, a genome doesn't have negative values. And when you're seeing a negative value, it's indicating that the SNP is occurring very close to the beginning of the contig.
So Rebecca, while you're doing that, there was a question from
Sure.
Sally. She's asking about, the fact that antigenic variation takes place in the outer membrane proteins. Can you actually remove them from the SNP analysis? Can you exclude them from the SNP analysis? I'm assuming you can filter specific genes from the table you showed us before.
Yes, you can filter genes from that and then download those. And also when you download everything in Excel, which I always do, that first table I showed you that showed you everything, it gives you the names of all those things, and you could just do a like a conditional format and look for outer membrane, because generally
Right.
they'll have that name, and then you can just get rid of those if that's what you want.
But you can't get rid of them. You can't mask them basically pre-analysis. I think, Sally, maybe that's what you were asking.
No,
Sure.
sorry, Sally, we cannot do that.
Yeah.
So in this case, I'm just going to add the 2,000 to the start. Sorry, I know this is.
And another question from
Yeah.
Lisa is why did you use plus minus 1000 again? Like what
Oh,
was that number?
it'll be crystal clear in a second. But the viewer that we have to view these things, if you give it a single nucleotide, it'll be too small because it's looking across. I can't remember how big this genome is, but you know, it's like looking at an individual house in the United States. It would have to be a lot bigger for you to actually see it. So you make the centroid or whatever to show it. That's not actually reflective of the size, but it's giving you a sense of where it is. Does that make sense?
Yeah.
Okay. And then here I'm just doing this to.
But in theory, you could just use the exact location and it'll just.
Yeah, but if I did,
But you have to zoom in. Yeah.
Well, no, you can't zoom in the viewer. This is just to create a figure
okay.
for a publication. It's
Okay,
simply
perfect.
that. If you want to see exactly where it is in the genome, that's what that genome browser is for. So that's the
Perfect.
drilled in view. This is the view that I would, if I wanted to put it into a publication, this is the view for the publication. So I'm putting the start code on here so it knows where I'm the start site here so it knows where to start. I know I apologize for this, but the downstream thing is really nice. So okay, so I have this, but in these I have a formula here, so I have to paste the values. If you guys use Excel, you know about this. So now it'll be these values instead of this, and so then I can get rid of the position because I don't care about it anymore, and I can get rid of this top column because I don't need that, and then I can save this as a text file, and I'll call it SE1 now because I've named it several ways. That exclamation point will probably cause problems, and it's saved as a tab delimited text. So yeah, and yes, we're agreeing that that's well. So I'm sure you're thinking, why did she make me sit through this? This is why. So I'm back to the feature page, but I want to go to the genome page here. So that was a breadcrumb at the top, so I can click on this. And this is the page about our reference genome, and you can see there are a number of tabs, all with different information about it. But we have a circular view. And when I look at all the papers that cite the resource, this view is I often see it in the papers. And so we have, this is the main chromosome, and these are each of the plasmids. And yes, it's a circular view. The other people did linear. I know some of us would rather have linear view of this, but this is what we've got. And the default shows you the forward strand, the reverse strand, RNA genes, genes within this genome that have blast hits to AMR genes, to virulence factors, to drugs transporters, and to transporters, and then to drug targets, and GC content and GC skew. One of the beautiful things about this particular viewer is I can get rid of all these. So I don't want that, I don't want that. And then I can add my own data. And that's why that format was so important. So I can choose the file I want and say I want to open it. And I can call it SE, and if it was for a publication, I would make sure it was the right name, which is probably A17. I'm not going to, this is just for the example. And then I can add that. And see that? See how that pops up? Those are the areas where the SNPs are occurring in that genome. And so you can see that, and you can see clearly, like this plasmid doesn't have any genes annotated on it, but there are a number of SNPs on it, and you could highlight it. And I'm going to have to pull this down a bit so I can show you. I believe it's here that I did it. No, it's here that no. It's hard when I've got this up. I'm going to admit that person. There we go. So I did this here, and you can see this is a different way from the way that the other paper was doing it, and they had the linear view of just the main chromosome. But here we have the forward and reverse strands, and then you can see along the side the different views that I have for this. And I can even go in and modify this to show different colors if I want. If I want the forward strand to be this, the reverse strand to be maybe a lighter gray, and then I really want these to pop, so I start bringing in the colors. And so you see how you can manipulate it to view it the way you want, and then you can also see who has the SNPs and who doesn't have the SNPs. Like some of them are, come here you. This is something unique to this guy, and it's telling you the exact location that I manipulated, but it's still the general area that SNP occurred. And so, going back to the slides real quick, when we do the result comparison, the authors, these are the different bioinformatics tools that they use. We have similar tools in BV-BRC, and then I went through and looked at these things, and I mapped it. I just did this myself, so I could see what the different plasmids were to give an idea if I was going to submit this as an article. It would, I would want the plasmids to be labeled, but you could also load the plasmids individually and run the SNP analysis against them, because they all exist separately, and the two different visualizations. So now I want to change gears. If there are any questions, speak now or hold your peace for a little bit, because we're going to go into the RNA-Seq analysis. Any questions?
I have a quick one in terms of, so you said that you showed the trimming, so part of the BV-BRC workflow is that it does some trimming, right?
Yes, you can do trimming. You would go over into, you go over here, I would go into, come here you, sometimes it gets a little persnickety in it. Once a refresh, as we all do at times. Sometimes we just have to step away. In the FastQ Utilities, I can trim, I can do paired filter. Sometimes people have unequal read pair sizes, and that will cause things like assembly to fail. And so this will even things up for you. FastQ C tells you about the quality of your reads and if you need to worry about them, and align. So if you wanted to trim, you would do this, and we use trim galore.
Great. And you can add your custom libraries, it looks like, because it looks like you can select libraries down there on the bottom, right?
You can.
That's
well,
great.
What you're doing is you're adding things to a selected library. So it would be your reads, your single reads, and then you would add them individually. Now, if you're interested in command line to be able to do these things without the interface, we do have that option too. So you'd go here to the CLI tutorial, and it tells you how to download the command line interface. So for the people who are not computationally challenged like I am, I do everything through the interface. But does anybody else in the project do that? Not so much. They all do command line.
So another question from...
Sure.
Oh, yeah. So another question from David Alred. So he's asking about whether the SNP pipeline also calls indels or short.
Sorry, I'm having trouble speaking coherently. Let's see, I think I had it up. Hold on. It's this stupid thing at the top. It gives you SNPs, MNPs. Come here, you. I'm going to show you really quickly, because I really like this pipeline, so I don't mind showing it. And it gives you large scale deletions, things like that, and insertions. So let's look. So like here, it's showing you that there's a big deletion. And here is an insertion. Now, Does that get close to what you want? If you wanted an inversion or something, I think you'd need to look at genome alignment, which is our MOV service, then that can show you at a larger scale chromosomal thing that's showing you those things. Make sense?
Yeah, it makes sense to me. And David and others, if you have more questions or you didn't get an exact answer for what you wanted, please feel free to clarify.
Yeah, you can just do help and contact us, and then we can just talk about it all day long until you beg me to stop. Okay. All right. Now for the second part that I wanted to go through, because this is new, and well, we've always had RNA-Seq, but we have the ability for you to do dual RNA-Seq as well in the resource, and I wanted to show you a couple of the new pipelines. Some of them are still in development, so I don't have the tutorials quite ready for it yet. The newest one that I'm so excited about, but I wanted to show you this. So this is dual RNA-Seq analysis of Orientia tsutsugamuchi, and I can already hear it. Oriental tsutsugamuchi is not transmitted by ticks, but it is transmitted by mites. So it's close, right? They're both arachnids, so it was really close. And this was a dual RNA-Seq example, and I was just so thrilled that I was able to do it that I wanted to show it to you guys too. So this bacterium causes severe febrile illness, and the authors looked at two different strains of Orientia tsutsugamuchi, which they started calling OT, because who wants to keep saying tsutsugamuchi all the time. Strain carp causes a very fast lethal infection in mice, even at low doses, and there's a closely related strain, UT176, that was isolated in Thailand in 2004. Now what they did was they took carp and they took UT176 and they incubated them with human umbilical vein endothelial cells, or HUVEC cells. And then as their analysis, they sequenced those samples, and then they took the reads that came off the sequencer and they mapped them to carp, and they mapped them to UT176, and they mapped them to the human genome. And then they did a differential analysis, two different ones, looking at the way that carp and UT176 responded at day 5 after infection. And then they also looked at the way that the human cells at day 5 responded to infection with carp versus UT176. So there were a number of samples, but sort of one sample, you could look at it two different ways. They trimmed using Cutadapt. They did mapping using Redemption pipeline. Actually, that's a very clever name. I hadn't really thought about it until then. And Segmel. They mapped to the OT genomes using Salmon, which does indexing and quantification. And then for differential gene expression, they use the EDGE-R package in R. And anybody who looks at RNA-Seq analyses know that basically it may be the law of RNA-Seq that you have to include heat maps, you have to include volcano plots. So they provided both for both humans and human cells and for bacterial cells. So how much of this analysis could we do in BV-BRC? And I just wanted to show you what my frame shift, or the way I did it. So I was able to take their reads and I aligned them to the carp genome and the UT176 genome. So when I aligned it, it gives me the mapped reads that I could put into two of our pipelines, Tuxedo, which is our old standard that we've had forever, and our new one, which is HT-Seq, that brings in DE-Seq2 for the differential expression. Now the mapped reads were to the bacterial genomes, but there was also when I use that particular service, it gives me unmapped reads as well. And those, I did RNA-Seq analysis using the host HiSat2 pipeline. So I'm going to leave the slides for a minute, and you'll be out of the dark in just a second. And then I want to show you exactly how you can do this. So let's first start with aligning the reads. And so we go into Tools and Services. We go into FastQ Utilities. I created a folder. So, oh, come on. There it is. I'm going to use that folder, and I'm going to call this Align to Carp. So I could do trimming, I could do quality. I just want to do aligning for this example. So once I choose my pipeline, I add it in, and you notice right away it says, hey, what target genome are you going to do? So I don't want to write all of Orientia. I wonder if I can get by just writing carp and see if it will give me that. And it does. It's looking through the database saying, what can I find that matches this? So this is the particular genome. You notice that we have two versions in BV-BRC, but this is the version they originally used. So I click on that. Now I have, if I had paired reads, I could put them in. If I had single reads, I could put them in, but I have SRR numbers. So I can put that in. You can see that once again it was from my Word document. And then I have to move this over. Come on, move over, don't be shy. And it was taking a minute. Then I want to make sure it's that. And so it's gone to SRA, and it said, what is this? And SRA tells me. And so I could enter them all, but I'm just going to enter this one right here. And I'm going to click the Submit button. And once I've submitted it, I can see that the job is running. And look, you can see that that variation job completed really quickly once it started to go. Oh, actually, that was a few days ago. This one's still running. But let's go into the workspace, and I want to go to public workspaces because I just want to show you what the completed job looks like. And then I go into BV-BRC workshop, click on that, scroll down to TIC webinars, click on that. And now we're in the RNA-Seq analysis with Orientia, so click on that. And let's look at reads aligned to carp. So click on that. Now remember, I did everything. I aligned the reads to carp and I aligned them to UT176, but I'm just showing you this particular example. So for these particular read files from SRA, so when it has the checkered flag, it means that job successfully completed. So we click on that. And you can see that it gives me, when the pipeline runs, it gives me BAM files. And it also, this is the beauty of it, it gives me reads in a gzipped file that aligned to the carp genome in this case, and reads in a gzip file that didn't align to the carp genome. These reads I'm going to use in the bacteria versus bacteria analysis. These reads I'm going to use in the host response to the two different bacteria. So these should be to the human genome, these are to the bacterial genome. And this just is going to give me an indication of how many reads there were. So I highlight that, I click on the view icon, and here's my little chart. And you can see, oh, 10%, only 10% mapped to this carp genome. But look at how many reads there are there. Actually, the number is more important than the percentage because there's a whole bunch of reads there. So this is going to be a nice analysis. And the unmapped that are going to the human genome, there are even more there. Okay.
And Rebecca, is there a reason why you did it this way? I mean, in theory, you could also just use the same SRA numbers to map against the human genome, right?
I play it you can map it to the human genome. You can also just use the SRR numbers in it without mapping it to anything. So you can do all of that. But this was more, actually at the time I did this, it was throwing an error on the human genome because we weren't, it was running out of time, which we fixed at the time. So this was my way to weasel around it. There is always a way. If you're blocked in something, what I found is I can always squeeze around it one way or another. You just think about it for a little bit, go out and get a cup of coffee, and then come back and say, oh, I can do it this way. I'm not going to be stopped by this. So that's my rally cry
great.
to you all. Okay, so the most important thing is, remember, we have the aligned read files and we have the unmapped reads files. So let's go do some RNA-Seq. So we go into Tools and Services and we click on RNA-Seq analyses. And we have 3 different pipelines, Tuxedo and HT-Seq DE-Seq2. Those are both for bacterial genomes. Host HiSat is for the human genome. So I need my target genome, and I'm going to put carp there, and let's hope, good, it finds it again. Now, remember that we have reads mapped to carp and reads mapped to UT176. They're both close relatives, but we're going to compare them both to this carp one to be able to chart when the expression is going up or down in either of these. We have our output folder, which is here, and I'll say reads v carp, and I will first submit the HT-Seq pipeline, and I always put the date on it too. And then this is you always have to enter stuff in here first before you can describe HT-Seq. Now you could just run these things and not do a differential expression, but what fun would that be? So I want to compare between the two, so I have to create conditions to allow the pipeline to say, oh, you want me to compare this versus this, I get it. And so I can name that condition. I'll call one carp for the reads that align to carp, and I'll call one UT176 for the reads that align to that. Now, I could, as Omar pointed out, I could enter the SRR accession number here, but I've already sort of gamed the system. And remember that there were a lot of bacterial reads in that file, but there were even more human reads. So if I do it this way, at least I'm eliminating all of the human stuff and allowing the RNA-Seq pipeline to concentrate on what I want it to concentrate on, which are those bacterial reads. So it was this SRR number, because now these are in my folder. These are in my workspace, so it's looking for them, and it says, oh, you have unmapped reads, and you have aligned reads, and this is in my workspace, and it, for some reason, it usually does that, and I can just do that and say aligned. And then these were carp, so I move this over. And then this is how I'm going to show you how you can run a bunch of jobs simultaneously. So 18, yes, move this over. 19, move that over. And now I have to change conditions because these are UT176. 20, move that over. I said move it over, and you can see that the little thing has changed because it says, oh, this is what you want me to compare it to. I got it. 21, move that over. 22, move that over. So we have our 3 things here assigned to the 3, and if you wanted to check them and check against your Excel spreadsheet to make sure you've got the right conditions assigned, you're good to go. And oh, I still can't submit it because I haven't done a contrast yet. So I'll do UT176 versus CARP. In retrospect, I should have done it the other way, but because I did it when I originally ran the job, it's here, and now I can submit this job. So it's submitted. Once it's submitted, I get the thing that says, I'm submitting your RNA-Seq job, it's been successfully submitted. But this is something I always do too. Once I've submitted it, because I have another bacterial pipeline, and I went to the trouble of uploading all of these dumb reads, now I want to use the other pipeline. So I'll say CARP versus UT176 tuxedo 14 June. So I have that, and then I just have to do the same things. And look, okay, so the first one was HTC. I went to all the trouble of loading that. Now I can do tuxedo. So that job's loaded and running. I always do that. I always make every comparison because I want to do what's best for me. That sounds ridiculous, but really that is, I want it to get the best results. So I'm trying all different things to see what works the best. Now we want to, while we're at this, we're going to submit another job, and the reference genome this time will be Homo sapiens. So it's searching, and there it is. There's the Homo sapiens unique identifier in BV-BRC, so I click on that, and notice that it changed here, and it says, okay, if you're doing Homo sapiens, we're doing host HiSat too. So I do CARP v UT176 host HiSat 14 June. But wait, we don't want to compare these, because these are the read samples that are mapped to the bacterial one. We want to grab those unmapped reads. So we have to say goodbye to these, one after the other. Goodbye, goodbye. And instead here, now we can do it and say unmapped. And so this was 22, and then 21, which was UT176, and then 20, move that over, and then 19, and I have to change the file here to the CARP to make the comparison. 18, unmapped, move it over, and then 17. Mapped, move it over. So now I have my comparisons. It's going to say, we'll do the same thing again, UT176 versus CARP. Do that, click submit, and this particular job, the RNA-Seq, okay, things that are really expensive and run really slowly are generally assembly. The trees can be slow if you have a huge number of genomes and genes that you want to do, and RNA-Seq is really slow. RNA-Seq, when you're looking at the human genome, it's going to be really slow. I believe this took days to run, but so they've run now, and we could check it on our jobs folder to see which ones are running, and I can see that one failed, but I'll check on that in a second. Could just be a system error, but I want to go to, because it worked for me before, so I have to, I'm sure it's just something weird that went on at the moment. So I go into BV-BRC workshop, take webinars, RNA-Seq Orientia. So first I want to look at this HT-Seq to E-Seq2 data. So let's click on that. And this checkered flag means that that job successfully completed, so I click on that. Now a bunch of stuff is coming in here. So I'm going to show you different things that you can do with each of these. You could look at things in the browser, but I don't care about that right now. What I first want to do is look at how I can look at the different expression of the genes. So to do that, I'm going to click on this checkered flag, which is showing me the differential expression part of the job. So I click on that. And then up here, it gives you to a page with a bunch of JSON files. Nobody wants to look at those except our hardcore programmers, of which I am not one. But up here at the top, we have the experiment icon. So let's click on that. And this is where we can start manipulating the results and seeing the data. And it says this is the one comparison that you made, which was UT176 versus CARP. And there are 2020 different results for these genes. I can go into the log ratio, and I can say, I want to see everything that's twofold expressed, and filter that. And then it tells me, oh, these are they. And you could even click on it and try to see what those were. And I could even say, I want to see the ones, because if they're up, remember I was comparing UT176 compared to CARP, and this is at day 5 after they've been incubating with those human cells. I want to see what's up in UT176. So I click the up, and then it says they're 171. So I can grab those, or I could look at it in the heat map, which is giving me some indication of how up they are. The more intense the red, the higher the change. And if you're things that you're more interested in, I can use my mouse and scroll over it, and I can tell it to show me those genes. So that's one way that I can look at that data. So let's go back. And I want to go back to this folder here. Another way, because you could see that there are some genes that are up and some genes that are down. This is something new that we've just added, and we're going to wrap this all into a report like we have for comprehensive genome analysis, so you can look at it in detail. But we have these report images, because what we do is we take that data for you and we try to summarize it in a way that many of the RNA-Seq papers that you read about do. So click on report images, and you could click on the heat map view. And this is showing you the differences between the CARP versus the UT176, those three things, and it's showing you the top 50 normalized differently expressed genes. This is the end of that gene identifier here. If it's red, it means it's higher in the TPMs versus 0, which means it's not so high. And then if these things are colored red, and I think, oh yeah, it was the last one that somebody asked about the membranes. Here we have it in the heat map, so you can see which genes are identified with membrane transport. So we have the heat map view. Clicking on this takes me back to that. We also look to see, are there any differences in genes that are known to be in pathways? So we can click on that. Red are for the CARP, the greenish ones are for the UT176. So it gives you a standard violin plot of those. And, you know, granted, nothing, when you look at this, nothing screams to you, oh my gosh, I have my Nature paper. But still, remember, we're doing RNA-Seq, and it's still pretty cool that we're able to do this. And it's just that, you know, not everything, you're not gonna get stuff that's gonna lead, there's a lot of searching that needs to happen when you're doing your own analyses. Come on. Okay, so this is looking at the subsystems, looking at the data around the subsystems, and it seems to be a little bit slow, and I hope it's not me. Are you guys all still there? I worry when it slows down.
We're still here. We're still here.
Okay, because my internet could be slow at home, so I thought, oh no, I've lost them all. But the subclass is just something similar, and then we also have a standard volcano plot. So we have the report images that you can look at. And then the last thing, well, there's a number of things. This number here, which we need to address, it's the genome identifier for the A1 Orientia Tsutsugamutsu genome. And so there are even more files in here. I know, you're thinking, gosh, it just never stops. But you could also use this and click on this one, which is the DE-Seq2 TSV, and click on View. And that will show you all the values associated across each of those genes so that you can look at that, and you could click on the fold change to look at the most highly up or down regulated. You could filter on the different columns and keywords. So that's for the HT-Seq. But we did some other pipelines too. So I want to go back to here, the RNA-Seq analysis with Orientia, and show you what we have for the host HiSat. And so those were those unmapped reads that I used and mapped them to the human genome. So I click on that. Now we don't have as much stuff with this because the human genome is so dang large that we don't have similar functionality, but we have some good stuff. So we could click on the report images, and well, I need to click on the folder, and you could see the top 50 different differentially expressed genes between when the human umbilical endothelial cells are exposed to carp versus UT176. So you could look for changes in those, and these are human genes here that you can't that aren't, well, I guess they are somewhat in the...
And Rebecca, these are simply based on fold change in TPM values,
Yes.
the transcripts per million. It's not doing a differential expression with statistics and so forth for the human.
Right, that's right.
Okay.
But we do have that, and I'll show you that in a second. So we also have the Volcano Plot, but this is so big, I'm not going to click on it. But I will show, what I would do if I were you is just download it and open it in like Illustrator, because it's gigantic. Okay, so go back here, and this is the unique identifier for the human genome. So I click on that, and then we can also look at the DE-Seq file and view that. And here is where you can see the actual values and the fold changes when you are comparing UT176 to CARP. So which human genes are up in one versus the other. You can download it, but there are a lot, right? Because the human genome's really big. One last
And
thing.
can you, just
Yeah.
a quick question, sorry, this is very nice, obviously. Can you do double sorting on 2 columns, or you'd have to download that to do in Excel?
No, right now you would have to download it in Excel. Like, this is new functionality, so it would be nice. In a lot of our other services, you can open a filter and filter on that, and so we could try to move for that. It might be nice to be able to move to that other interface that we had for the bacterial genes, where you could filter on fold change and keywords and this and that and the other.
Yeah.
But we don't have that for this yet. So those are
In
all
this case, obviously one would want to filter or sort on fold change, but also sort a second sort on the adjusted p-value, just to see what is actually significant out of the ones that are differentially regulated.
So,
but you can do that in Excel, which is not a problem.
Yeah, you can always do it in Excel, but it's a big Excel file too, with all of those genes. Just one more thing that I wanted to show you that I didn't show you last time is we also, when you do the RNA-Seq, it gives you a analysis of the statistics of your reads. So just so you know that that's there too, so you can look at that if you're interested in that kind of thing. So there's one last thing that I want to show you. And we're leaving Orientia, but I wanted to show you when you have more in each of these. I've only had one contrast of one to the other, but I wanted to show you some of the value when you have multiple contrasts. So I loaded this particular job in there that I did a long time ago. Well, not too long ago, but here's the checkered flag indicating that it's done, and I need to go here to see it, and then I want to go into this experiment, and you can see already while it's loading that there are a lot of comparisons that I had there versus control at 30 and 60 minutes, and then looking at the treatment of what those are. And here, this is a salmonella genome, and I can say I want to see the highly differentially expressed things and filter on that. And then I'm down to 1,165, but I'm bringing this up just to show you what you can do when you get the heat map here and you open that up and you think, oh my gosh, how am I supposed to find anything from that? When you have more than one treatment in comparison here, you can cluster that data. And then things start to pop out and you can see which genes are going up, which is red, or down in green in different conditions. And then once again, you could grab those and then show me those genes. And so you can see that kind of data and it'll load it, that kind of thing there. So that, I mainly wanted to show you that heat map view of things when there's more there so that you can compare it. So it's a quick end because I know we're late, and the result comparison, just the different bioinformatic tools between the two analyses. They had heat maps and violin plot. They had heat maps and volcano plots. We had heat maps and also some violin plots. We allow you to filter on genes of interest in the bacteria. We have images from the host HiSat. This is the one that was so darn big. And we also allow you to filter on the host data. And I know I've run over, but do you have any questions? Or has everybody passed out?
No, that was really great. So maybe we'll give people a minute or two. If they have questions, feel free to unmute yourself or just type it in the chat. Otherwise, obviously, the recording will be made available very soon. You'll get an email, and anybody who didn't attend will get an email with a link to that recording. Definitely feel free to view those and share it with your colleagues. And Rebecca is always more than happy, well, anybody in either BRC, but in particular for this webinar, Rebecca is more than happy to answer specific questions now or later by clicking on the help or the contact us link and sending an email.
One other thing I can show you really quickly is when something failed, you have a number of things that you can do. Now, if I wanted to see why it failed, I could go in myself and look at the error message, and I could see, oh, something weird went on. But I could rerun it if I decide that that's not a valid issue. But what I think everybody should do is report it, and this will send, you don't even have to say anything here. The job failed, you submit it, that comes to me, and we have something that the programmers can look at to try to see why it failed. And then they can say things to you like, ooh, your read pairs are unevenly matched, so you need to go into FastQ Utilities and do the pair-to-line function, something like that.
There's a question from Dina Amin about she's just asking how many replicates. I'm assuming, well, this could be two types of questions. One is how many replicates can you actually load? And I'm assuming that's limitless. But the other
will it run? Every job that we have, generally we try to allocate it an amount of time and memory. So we try to average things. But if people are loading really big things, I'm trying to remember how long that human host thing went, and so if it outruns it, the jobs will sometimes fail because it outran the time allotted to it. So the more stuff you put in there, you're playing, like this ran 37 hours for that human genome, and it was still completed, right? It didn't fail, and as we get more data, if we start getting a ton of these stuff going in and mapping against the human genome, then we will start allocating more memory for it because we'll have more experience with it. But that's why you should report a job if it failed, because often it's that it ran out of memory. And so the developer looks at it and he says, oh, it ran out of memory. You know what? I'll give it double the memory, and I'm rerunning it for you. So you can make it as big, but after, if you're too unreasonable, if you have 1,000 things to compare in this. First of all, how are you going to compare it at the end? Maybe running machine learning on it to figure out
Right.
how to analyze it.
But in terms of how many replicates you need, I mean, I think the standard right now is you need 3 replicates to do.
Yes,
But
you
I've
do
seen
need. Oh, in HTC2, you need 2 replicates to run it and get differential expression. If you only have 1, it won't run it. But 3
right.
is
So
generally the standard.
yep. But you can do it with 2. But the recommended standard is 3. Obviously, the more replicates you have in theory, the better the statistics are. But I think
Right.
you reach a point after 3, 4, or 5 that the statistics don't change that dramatically.
And the chance of the job failing increases
Exactly.
substantially.
Good point. Any other questions from anyone? And obviously feel free to unmute yourself if it's easier than typing.
Well,
All right,
this was
looks like we're
fun,
good.
guys. Thanks so much. And, any questions you have, let me know. And the tutorial for the new pipeline should be available later this week or early next week. But in the meantime, contact me if you have any questions. And thanks for joining.
Great, that was very nice, Rebecca. Thank you. There are lots of thanks from everybody for you. We'll talk to you soon. Okay, bye,
Okay,
everybody.
bye.